All right. Welcome everyone to the Points on Product with Brian Dixon, our VP of Product Development. Super excited to have everyone. Thank you for taking time Wednesday afternoon. I know it's a busy time, but I think there's tremendous amounts of knowledge here. I think we've proven that week after week. Brian, you've done such a great job in really breaking things down, giving us a different vision, a different mindset, a different way of looking at Protandem and all the ingredients in our different product line. And we've really kind of gotten really heavy and focused on the peer-reviewed studies. And it's interesting, there's a lot of amazing distributors on this call today. And I want to just bring up one. Michelle Scaff and I have had a lot of different conversations. And one of the things that really stood out to me in one of those conversations was, back in the day, back when LifeVantage was first getting started and these peer-reviewed studies started coming out and they started hitting, having an effect on LifeVantage. Obviously, there was a tremendous amount of curiosity with third-party validated, type institutions. They were like, holy cow, what is this Protandem? What is this product? And obviously, a lot of those peer-reviewed studies started hitting, PubMed and all those different things that gave us a better insight. It gave us bigger arrows in our quiver to go build our business, tell the story, and obviously increase the distribution or the consumption of this amazing product. But what stood out to me, Brian, was, Michelle was saying how interesting and how much it fueled the field of the distributors of LifeVantage in such a way where it really empowered them with knowledge of exactly what the peer-reviewed study meant, what it was, why was it working the way it was in the body. And so now we've gone back to that and we're starting to see tremendous traction. If you look at the YouTube channel on the Vision Summit, if you go just, Google or go to YouTube, search Vision Summit, and you'll see all of the recordings that we've done week in and week out. Dr. Dondi Nettles, a Pro 9, has done a fantastic job. And Brian, we actually started communicating, Dondi, myself, and you, with regards to kind of just having this three-headed monster. Dondi has a brilliant mindset as a doctor. I'm just an entrepreneur that doesn't really understand a lot of this science. And then we've got your brilliant mind and heart with regards to product development. And so I think all of us playing off of each other, it could be pretty comical. It could be pretty deep with knowledge. But overall, I think we're giving a tremendous value add and benefit to those that are looking at the recordings or listening to it live. So we've got an amazing peer-reviewed study that we're going to break into. And for those that don't know, but we are going to get into, hold please. I wrote this thing down and I don't know if I'm even saying it correctly, but it's called,
this will be fun. Are you going to read the title?
Yeah. Hemo oxygenase. Is that it?
Heme oxygenase. Yeah.
Heme oxygenase. And the importance of what that is. And so, Brian, I'm going to let you do what you do best, man. We're so thankful for your time, your vision, your passion, your consistency with this call. We are our benefit to the world. And so I'm going to turn it over to you, and then we got a bunch of questions for you, and we'll get into this. So, Brian Dixon, take it away.
Yeah, sounds good. Thanks, Fry. And quite frankly, though, I would take your entrepreneur paycheck versus my doctor paycheck. Just saying. But
me
anyway,
too.
So yeah, let's jump right in. And, like Fry said, this one's dense. This one's full of molecular biology. That's my wheelhouse though. So I love it. And I did throw together a bunch of slides to kind of give you all some background on some of the things that they're talking about. But just to kind of jump right in, right? You guys know I like to start with the title. So synergistic induction of heme oxygenase 1 by the components of the antioxidant supplement Protandem.
Hey, Brian.
So it's so interesting. Yeah.
For some reason, we are not seeing that screen.
Oh, really? You don't see the paper, hey?
Nope. So maybe just stop screen share and then try it again. Let's see if that fixes it.
Look at that. Golly, it's green boxed on mine. Are you still not seeing it?
Yeah, still not seeing it. Maybe instead of it going into video mode, just keep it outside of like the kind of the slideshow, and maybe it'll pick it up then.
Yeah. What about just for fun? Can you see that keynote presentation?
I'm not seeing anything. Guys, are you seeing that? We're just seeing a black screen, my friend.
Yeah, maybe we should ask Steven. You can see it.
Yeah.
Oh, shoot. I don't know what's going on. I can see it on my screens.
Can you? I'm wondering if you're sharing it on are you on a Mac?
I am, yeah. It hasn't been an issue.
Is Brian set up as host or co-host?
He is. He's a co-host. I could make him host.
No, it shouldn't make a difference. Is it a doc, Brian, or is it in?
Yeah, it's I have I have the document, the PDF, and the presentation, kind of like we've done in previous weeks.
Because I can't. Sometimes I can't show it just a doc when I'm trying to share a screen. I don't know why, but anyway, I don't know.
Yeah, and that's why I tried.
Hey Brian, when you click screen share, does it take you to options on like you know what that actually is, like home page or different visuals?
Exactly.
And you're clicking on the home page.
Still can't see that, hey? Why don't let me log off and log back in. Right,
okay.
Right, sorry. Or do I call you Patty for today?
You can call me Patty. I've been called much worse, and Patty just elevated myself. So you could call me Patty all day long. I love that woman.
And Brian, close everything first before you log off and then go back in again. So.
Okay, I'll give that a go too. Thank you. All right, I'll be right back. Sorry, everybody.
Okay, no problem. So guys, this is a really interesting conversation today on hemooxygenase and why that's important with Protandim, but really how the phytochemicals that are a part of Protandim and how they play into this whole benefit for the body, the mind. And Brian will really get into this. And I learned a lot of new things with regards to why it's so important to have all of the encompassing ingredients, the key ingredients that make Protandim what it is. It's not just one key ingredient. And this is kind of a really cool peer-reviewed study that kind of proves the fact that you could take one of those key ingredients and it really didn't have the effect. When you add all of the key ingredients that we have in the Protandim, amazing things start to happen. So we're going to get into that as soon as Brian logs back in. Thank you for being patient. I like this, gave us extra time to get others on live. I like that. And then guys, if you have any questions with regards to that, just put it in the chat. I'm going to be watching the chat. I'll make sure that we ask those questions and so forth. For those that we don't get to, then we will submit them into the questions on product and make sure that those questions are answered for you as you guys continue to move your businesses forward. See if Brian can get back in here. I wish I had more knowledge like Dondi did. I mean, I know Dondi had like a 10-minute break before Brian came on, and man, that was like listening to another keynote speaker. So I apologize, I'm not brushed up to speed on that kind of insight and knowledge. But if we were to talk about network marketing, game on. I've got about 8 hours before I take my first breath. So, all right. Let's see. Maybe he got lost.
Yeah, share with us what you know about hemooxygenase or whatever it is.
Man, I have no clue. Steven, no clue. I've never even heard of the name and what it does, but I'm excited to learn about it. That's for sure.
I'm going to tell you a little story. When I first got started and this came out, I shared this one study with a good friend who's a scientist and a researcher creating drugs in his lab. And he looked at this and hemooxygenase 1 was like the big thing for him. Are you kidding me? This is anyway. So the whole thing with hemooxygenase 1 and the importance of it for our health is everything. And I won't go into what he said because I would ruin it. Brian's back though, I think.
No, thanks for jumping on, Michelle.
And I love that he's covering this one. So I can't wait to hear what he says.
I know. Brian, we got you, buddy. I just made you a co-host. And for some reason, you're muted. Let's see.
Could you see that paper?
Nope. We just got a black screen, buddy. So I wish I could teleport right to where you are.
I know, dude. It's green on my screen, like it's sharing, it's highlighted right. I'm not plugged into another monitor.
Curious, could you send me that via email, and I'll see if I can just, while you're chatting through it, I can grab it and then share my screen, so forth.
Yeah, maybe. I did email it to you earlier.
Okay.
Right.
Let me pull that up 1 second.
Oh, golly.
Are you just pulling up the study, right? Is that right, Brian?
Yeah, but then it's a bummer. I threw together some slides as well.
Okay. So what I can do, let's just see if I can get this handled.
What about now? Are you seeing anything?
No.
No.
Oh, man.
It's okay. We'll get this figured out. 1 second. Let's see. All right. I'm going to share my screen, Brian.
Yeah, I got to think this through, right, Fry, because.
You guys can see that, right?
Yeah.
Yep, we can.
Okay. So Brian, you could just let me know. I can move it up and down, but I can zoom it in.
All right. Let me, right, Fry, let me also email you this presentation I've kind of put together as well.
Okay.
I don't know. Are you on a Mac and Keynote, I hope?
Yes, I am.
All right. Hallelujah.
And it's just my full name, Ryan Fry 2012 at Gmail. This is what I love about these calls, though. Like guys, this is real life, right? Like there's times where you can be so polished, so buttoned up and so forth. But I love the fact that, people can see how and why we're doing what we're doing. And there's times where it just doesn't come together just flawlessly and beautiful and seamless and so forth. Like, this is real life. So thanks for being patient. And I know the message we're going to deliver will be very impactful.
Right, Fry, if you could close your tabs and then also click on the X down there to close that bottom bar. That'll give us more of the screen.
Okay.
Over to the far right. There you go.
Close that. And then let's see, what did I just do? I could just get out of these, I guess. That's the best I can do.
Cool.
And then just let me know when you've got that, Brian, and I'll click over. Some people.
Yeah, and then I've sent that presentation to you as well, right?
Okay.
Let me try to share one more time. Nothing?
Nope, I'm not seeing anything.
And Brian, what are you clicking at the bottom when you go to share screen? There's two boxes down there to optimize and.
Yeah, I just go to share screen, Stephen, and then I select the pane that I want to share and then hit share. Like I said, I can see the paper. It's green box like it always is for Zoom. Well, I just got a message. Can you see it now? It says participants can see your.
No, I'm not seeing it.
Oh, again.
Okay.
All right. Well, we'll go with you sharing, right, Fry? But I'll have you kind of jump back and forth between that presentation and the paper, if that's all right, especially when we get down into the figures.
Absolutely. 1 second. Can you guys see that okay?
Yeah, exactly. Cool. So yeah, we'll jump back to the paper, and then we'll start working through. And I labeled each slide with the right figure, so you can kind of just see where we're at. But
okay, let me get back to, you want to start at the
yeah, the paper. That's all right.
box. Let's see.
You're gonna be busy, right, Fry?
Yeah, it's okay. All right, we're ready.
Cool.
I don't see the man, so we're fine.
I don't see the paper now.
You don't see that?
No.
Oh my goodness.
Oh my goodness. There we go. All right, like we were talking about, the title, the synergistic induction of heme oxygenase 1 by the components of the antioxidant supplement Protandim. And you know, when I was jumping back in, Michelle, I heard you talking about how important heme oxygenase is, and it is definitely incredibly important. Its normal role inside of our bodies is to basically break down red blood cells. So as red blood cells are running the end of their life, heme oxygenase is specifically handling breaking down the heme, or the iron containing component that's in our red blood cells. And it turns out that that's an incredibly dangerous thing to do, because our cells don't want free iron floating around, because it can be a free radical and oxidant generator. But in this publication, they were using heme oxygenase more as a surrogate for NRF2 activity. So we know that NRF2 will regulate hundreds of antioxidant and detoxification genes, but heme oxygenase and a couple others that they mention in this paper are really very well studied in their induction by NRF2. So in this paper, they were looking at heme oxygenase, like I said, as a surrogate for NRF2 activity. It's in the Journal Free Radicals of Biology and Medicine, a very prestigious journal, impact factor of 7.37. So a good publication. Looking at the author list, you know, I don't recognize any names on there except one, and that being Joe M. McCord. So he was involved in this study. Remember, not primary author, not senior author, but he's kind of there in the middle. So he might have taken a little bit of a back seat for the study, but definitely involved in the study somehow. Then again, if you remember the main components of a scientific paper: abstract, introduction, methods, results, discussion, and discussion/conclusion. The introduction is, it's pretty interesting, and it does or I should say the abstract does a pretty good job of really kind of setting everything up, right? So it's got, again, a little bit of an intro to start. Then it gets into the experimental questions and the methods. And then it also talks about, you know, kind of the second to the last sentence there was a significant increase in levels of total glutathione in Protandim treated cells. And I should also say one of the main reasons I chose this paper is I think we've probably all heard in the LifeVantage lexicon that Protandim can increase glutathione by 300%. So this is the study where that comes from. But notice in the abstract, it doesn't say by 300%. So this is a case where if you didn't get into the entire paper, you might miss that really pretty spectacular finding, actually. So we'll kind of dive right in. That first paragraph of the intro is interesting because it's saying oxidative stress plays a significant role in the progression of many diseases, including diabetes, Alzheimer's, and atherosclerosis. You know, those diseases are important to be called out here. One, because they're looking at heme oxygenase, but then two, also the specific cell lines that they chose. And we'll talk more about that when we get in to the figures. What else is interesting is they reference one of the papers we had done a couple of weeks ago, showing that Protandim can decrease T-bars within 5 days and increases SOD and catalase up to 120 days. So they're referencing that in vivo data in humans. And now what they're trying to do is get really mechanistic. So trying to figure out exactly how Protandim is working to activate these antioxidant enzymes. And this is where they'll use a lot of those molecular biology techniques to help answer that question. And the reason that's important, because in the human study, they gave Protandim and then they measured a physiological endpoint. So with the molecular biology, they're trying to figure out everything that's in between the giving people the supplement, looking at the clinical endpoint, but wanting to know exactly how that cell signaling is working in between those two events. So, Rye, if you could go down to the next page, kind of at the top half. So as we get towards the bottom of that second paragraph here, up just a little bit, right? That second to last paragraph in the introduction, it sets up the hypothesis where they predicted that Protandim is likely to be a strong inducer of heme oxygenase 1. They abbreviated HO1 because it's a phase 2 enzyme. So these antioxidant enzymes that NRF2 regulate scientifically are known as phase 2 detoxification enzymes. And then talks about how it's working and that breakdown of heme ultimately to carbon dioxide, iron, and bilirubin, a breakdown product of heme. And then right into the experimental question, the objectives of the present investigation are to develop a cell culture model to characterize the mechanism, so how Protandim is working, and this synergy between those 5 different components in Protandim, to be able to determine if Protandim induces HO1 in a synergistic manner through activation of multiple signaling pathways. So what they're saying there is, again, looking at the mechanism by which it's happening, and then what is the specific signaling pathway that's going to be involved. The other thing I should mention, we talk about the synergy of the 5 nutrients here. So this is one of the first papers that starts to show the individual ingredients in Protandim aren't necessarily that good at inducing either HO1 or NRF2, but together you see this massive synergistic effect. So Rye, if we go down to figure 1, we can talk a little bit more about that. So this is some pretty cool molecular biology that, again, if you're a little bit trained in science, it's not that difficult to do, but some pretty interesting techniques. So Rye, if we can go to that PowerPoint presentation and just start with that first slide, and kind of walk you through what this experiment's all about. Okay, so what is happening here is they're using what they call a Rhinella luciferase assay, okay? So Rhinella is a species of coral that lives in the ocean, obviously, but it has the capability to bioluminesce. It would be very similar to, say, a firefly. So there's also firefly luciferase, but they, in this case, chose this particular species to be able to look at the light that gets emitted. So they're using a gene from this organism to be able to measure the activity of HO1. So, right, if you can go to the next slide. So here, this will get a little bit technical, but I hope we can walk it through, and don't hesitate if there's any questions. Right, can you go to that next slide?
You here?
I still see the same first slide. Oh man.
Steven, do you see my slides changing? All you see is Rhinella?
Yeah.
1 second. Let me do this. Okay, can you guys see this now?
Yeah. There we go. So yeah, we can work off of this view, right? So remember, this is how NRF2 is normally working, right? It's normally bound to keep 1, and then something comes along, stimulates it, releases NRF2, it goes into the nucleus, and it binds to the DNA of these hundreds of different genes. What we haven't really talked about is you can see there, and what's depicted as the nucleus, you'll see some DNA down there, and you'll see something that NRF2 is bound to called the ARE. So the ARE is and it stands for the antioxidant response element. And so this is well characterized that this is the specific sequence that NRF2 is binding to. So if we can go to the next slide, right? So this kind of depicts that just a little bit more closely and in a pretty generic way. So you have NRF2 binding to that ARE, and then it's going to turn on those what we call target genes of NRF2, right? Those hundreds of different genes, and you get those genes activated and turned on. If you can go to the next slide, just to maybe orientate yourselves a little bit with DNA. Remember, DNA is made up of really just 4 different molecules, right? Thymine, cytosine, adenine, and guanine, or TCAG, they're usually abbreviated with. And the reason we can do some cool genetics and molecular biology is because of a base pairing rule that they call it in DNA. So C's and G's always bind together, and T's and C's always bind together. And you can see that depicted in kind of that cartoon. If you match up the colors, the blue and the green are always bound together, and the yellow and the red are always bound together. And that occurs billions of different times inside of our DNA. But you can obviously alter the sequence in literally an almost unlimited fashion. So based on how that sequence is working, or how it's actually laid out, will determine the gene, or in this case, kind of the promoter sequence. So just to get a little bit more technical to show something, if we can go to that next slide. So this is the actual sequence of those ARE sequences. And so when NRF2 is binding to this ARE, right, at the promoter or the switch to activate these genes, it's really nothing more than chemistry that's happening. So NRF2 will dock to a specific sequence within our DNA. So you can see there in the red is the core sequence of the ARE there. So now we don't have to go back to the figure just yet, but in thinking about that figure, what they did, if we go to that next slide, what they've done is taken the enzyme that's responsible for the reaction of that coral light, or if you want to think firefly type of light, this is what they've done. So you can see down there in the bottom left, it says RLUC. So that stands for luciferase. And anytime you hear ACE related to a protein, that means it's an enzyme. So this luciferase enzyme is chewing something up, and as it's facilitating that chemical reaction, light is given off, and we can measure that light. But what's really cool in this study, and where they did a little bit of genetics combined with that molecular biology, is what they've done, where you see that kind of triangle at the bottom of that. Yeah, exactly right. So down at the bottom, what you can do is cut this small piece of DNA, and you can put any sequence in this, they call them plasmids or vectors, that you want. And again, really, really easy to do. So we can take whatever sequence we want, put it in front of that luciferase enzyme. And in this case, if we put the ARE in this plasmid or vector, and NRF2 is having activity, it will drive that gene to be turned on, just like all of these antioxidant genes. And then you add the final component that that enzyme is working on, and you can measure the light. So if we go down to the next slide, this will depict it maybe a little bit better. So the promoter region, right? Think of that as the switch. So this is the ARE. And if you have the right activator there, you will turn on that gene in that plasmid. And like I said, you'll get light. So if you go to the next slide, this is what those different reactions look like. So focus your eyes down on the bottom. this bottom left, that molecule is what you will have to add into the reaction that Rhinella luciferase oxidizes to ultimately give off light. So what they're trying to do, right, just to reiterate, is they have the luciferase gene, they put the ARE as the promoter to see if there's any NRF2 activity, and then they're just simply going to measure the light. So go on to that next slide. And that's exactly what you can do. So if there's activity in your cells, you have them in these little, they call them 96 well plates. So they're like little mini test tubes going around, and there's 96 wells, why they call them a 96 well plate. So you can grow your cells in there, do your experiments, and measure the light. You just simply put it in a machine, and it will measure the light that's coming off. Didn't you go to that next slide there, right? Okay, we'll save that for the next figure. So if we can go back to figure 1 in the publication. This is exactly what they were measuring. So they had 2 different cell lines. They have that Min6 cell line, which is a cell line that's derived from the beta cells of the pancreas of mice. And so remember, they were talking about how oxidative stress plays a role in diabetes. So they chose a really good cell culture model to be able to look at this NRF2 induction. And then in panel B, they looked at a cell line from human nerves, so a human neuroblastoma line. So remember, they were also talking about Alzheimer's and some of these other brain diseases. So in my opinion, 2 really good choices of cells to ultimately be able to answer their experimental questions. And I really like how in this paper, they also looked at 2 very different cell lines from 2 different species. And the reason that's important is if there's a true biological effect, you should be able to see that across different cell lines. So what they did specifically is looked at the protandem formula altogether, or they looked at the individual components of protandem, right? The curcumin, the silymarin, think of that as the milk thistle. Silymarin is the active ingredient in milk thistle, bacopa, ashwagandha, and green tea. And then they increased the dosages. And so you can see at the highest dose in panel A, there was a little bit of activity with curcumin alone, but no activity with silymarin, bacopa, ashwagandha, or green tea by looking at that light that's being emitted. Whereas down in panel B, they see that curcumin in this cell line was a little bit more active, but still no activity in those other 4 components alone. But the other reason I like this figure is because of the dose response. And in biology, you almost always assume that there should be a dose response if you're having a true biological effect. And what I mean by a dose response is as you increase the dose of something, you should see a greater effect. It's less likely to be a random effect. And especially when we look at those top lines in each one of those figures, A and B, we can see just a beautiful dose response, especially with that final protandem formula. But we also do see it obviously with curcumin. So some good insight that this is a true biological effect. These compounds can induce that activity through specifically NRF2 because they're looking at that ARE promoter sequence. Does
Brian,
that make sense?
Yeah, absolutely. And Brian, like this right here substantiates exactly what this is stating here, right? Like where it talks a little bit about the objection, the components of protandem, and then it talks about right here, the protandem were tested alone at low doses. Curcumin showed minimal induction, whereas the others weren't able to induce it. But then it says down here, these findings suggest that the use of a combination of the phytochemicals may be an efficient method for the induction of antioxidant enzymes. And that's really what this is proving, correct?
That's exactly right. Yeah. And then, so this is really the major take-home message. And then they've done some other, they're almost control studies to really prove that it's a real effect. Because obviously this is an artificial system, right? We have synthetic DNA in there. We've had some genetic modification that's also gone on. But the rest of the study is really just trying to prove this figure correct. So if we go down to, excuse me, figure 2, this is where a lot of that synergistic effect comes from.
Wow.
When we talk about it. So here, what they've done is, instead of making synthetic now DNA, they're actually going in and measuring what's really happening inside of these cells. Right, if you could go back to that PowerPoint slide for me, I'll
Sure.
kind of walk through some background of how this study went down. So if you remember from your basic biology, and I don't expect many people to, it starts with, you have your entire DNA there up at the top, and then just a small segment of that has been opened up where an actual gene will be. Then you see that enzyme there, RNA polymerase. It will go just like a typewriter and work its way down the DNA, and it will make a mirror image copy of the message that's contained inside of that gene. And so that message is called exactly that. It's called messenger RNA, and so, or mRNA, that's depicted on the screen. And then that mRNA goes out of the nucleus into the cytoplasm, where it gets read by effectively another set of enzymes. And as it works like a typewriter down that mRNA, that's the code for the cell, or in this case, the ribosome, to keep attaching different amino acids to the string. It's the triplet code, if you've heard of that. So that enzyme will read 3, put an amino acid, move to the next 3, then depending on that sequence, we'll put that specific amino acid. Then the next 3, the next amino acid, and then you get this chain of pearls that ultimately folds up into the 3-dimensional structure. So each of these are very unique, again, because of the sequences that are there. You can vary those A's, T's, C's, and G's in literally an infinite order. And so depending on how they're scrambled, that's how you ultimately get that amino acid sequence in the protein. What is cool about genetics is you can work forwards or you can work backwards. So if you know the sequence of a protein, you can work backwards to figure out the sequence of a gene. If you know the sequence of a gene, you also know the sequence of the protein. So it's that specific in its instructions. But where I want to draw your attention is kind of that middle mRNA sequence. So once a gene has been activated and that mRNA message gets made, you can measure the activity of the gene by measuring how many copies of that mRNA are around, okay? So we'll still stay on this slideshow, but what they're doing in figure 2 is looking at the activity of genes by looking at that mRNA message. Okay, so right, if we go to that next slide. And the way that they do this is through a technique that's called PCR, or polymerase chain reaction. And PCR, or polymerase chain reaction, is nothing more than being able to amplify DNA. So you can see over there on the far left, we started with a single strand of DNA or RNA. You put it into this reaction, and there's 3 steps involved in PCR, and those aren't so important. But again, the whole goal of PCR is to make more DNA, okay? So in theory, 1 copy will lead to 2 copies, and that's depicted here, right? At that extension step, you see that 1 that we started with on the far left ultimately gets made into 2 strands. And then at the end of the next cycle, or the second cycle, those 2 strands have been made into 4 strands, right? Then the third cycle, right, from 2 to 3, 4 strands now becomes 8 strands of DNA. And so you get this exponential increase in the amount of DNA that's there. And again, remember, this is all very sequence specific. So Rye, if we go down to, well, let's stay here for one quick second. But what we can do, and the technique that they utilized in this figure, is what they call quantitative real-time PCR. So there's a technique that you can utilize, again, utilizing either light or a fluorescent probe, where you can measure how much of that DNA is being made after each one of those cycles. And typically in a PCR reaction, you will run this for 30 to 35 different cycles. So you'll see we'll get a massive amount of DNA by the end. So if we go down to that next figure, this is exactly the technique that they utilized in this paper. They utilized a fluorescent probe that after every cycle, you can measure the fluorescence. So as more and more DNA gets made, that fluorescence or that light will get brighter and brighter and brighter. And it's a little bit backwards, because if you look down at that cycle number, when there are the most copies to start with, you'll see the light come up most quickly. Whereas when you see really low copy numbers, it takes a while for that doubling to occur so that we get an appreciable amount of DNA in our reaction. And if you go one more slide, right, and we'll go back, this is exactly what they were measuring here in figure 2. They're looking at the amount of mRNA from a specific gene, right, in this case, HO1, that was made. And so the more copies of that mRNA that are around, the more quickly we're going to see that signal come up. And then you can back calculate to the actual starting concentration or how active that gene is. So does that make sense? I hope, Ryder, do you have any questions?
No,
there?
It makes sense to me, man. I'm just deeply following and just, kind of visually looking at it. But no, it makes sense, and it's hugely important. So let me ask you, like on this section here, the more that gene is being used, the more this is being mirrored.
Yeah, there will be more copies of that mRNA. So it usually for a lot of these phase 2 or NRF2 driven genes, they're really either off or on. There's not a whole lot of middle ground. So if that gene's on, you can expect there to be a lot of that mRNA kicking around. If the gene stays off, you wouldn't expect to really see any or much, if any, at all. So, right, if we go back to figure 2 in that publication, I'll walk through and just keep that mRNA message, right, in the back of your head and as a surrogate for the activity of the gene. So again, they treated, right, control, so probably just something like saline or salt water. And then they looked at the individual, sorry, right, go to figure 2 if you don't mind. It'll be down. Yeah, right there. So here what they did, right? They have the control, that light gray bar at the left for, again, both different cell lines. You see that's going to be what they'll call the basal level of the gene that's turned on. So nothing's been added. That's just the activity of the gene. Then they look at each one of the individual ingredients, ashwagandha, bacopa, green tea, the milk thistle, and the curcumin or turmeric, and then protandem as a whole. So we see something similar here that we saw with that luciferase assay from figure 1, where the individual components don't have a lot of activity, and we don't see that HO1 gene actually being activated until we get down to curcumin in the human cell line. But then very clearly, when you look at the combination of those 5 ingredients together, the PR denotation there, you see that massive synergistic effect occurring. So the individual components are weak inducers at best, but sure enough, when you put all 5 together, you get this massive induction. And then they start to try to figure out exactly how is that working. So just to take it one step further, they wanted to look at the actual HO1 protein that's being made. So, right, if we can go back to that PowerPoint presentation. Thank you. Go up one really quick, right? So remember, this is great because first they looked at the mRNA, so to see if the gene was turned on, but then it's also important to know, was that mRNA message actually made into protein? So then what they've done is they've went into their cell lines, they've ground up the cells, and they're just looking for now the protein HO1. So they're really covering all their bases in what it takes to make the final protein or enzyme. And the method that they used, if you can go to the next slide, The method they use is what they call Western blotting. So at the top, you can see a little petri dish where they've grown the cells. You'll grind up the cells, right? What they call the lysate there. And then basically add that lysate to the top of a plastic type of gel. And we talked about this a couple weeks ago. That plastic gel is acting like a little matrix that the protein has to wiggle its way through. So what they'll do is put a positive charge on top, a negative charge on the bottom, because most proteins are negatively charged, just like magnets running away from each other. You start to push these proteins from all of that ground up cells, right? And there's going to be tens, hundreds of thousands of different proteins there, right? All of these proteins, when you turn on the power, they start wiggling their way through that gel, and the smaller proteins are able to wiggle through faster than the larger proteins. So you're separating them by size. And then you transfer the protein that's in that plastic gel over to a membrane where you can do some pretty sophisticated molecular biology, and utilizing especially antibodies like it's depicted at the very bottom, right? Antibodies are very good at recognizing different proteins. That's how they work in our body. If we're infected with, let's say, a bacteria or whatever it might be, the way antibodies work is they're recognizing a protein that's most likely on the surface of a bacteria. Okay, so that antibody binds, they're incredibly specific, and they irreversibly bind as well. So what we can do is make antibodies specifically to the protein we want to look at. In this case, it's HO1, and we'll incubate this membrane now in a bath of these antibodies, and then we can visualize that again utilizing light. So if we can go back to the paper, this was, and if you notice those lines that are on there as well, what they've done is they've done exactly that. Down one more figure, right? So what they've done is ran those proteins down a gel, transferred it to a membrane, incubated it with antibodies, and looked to see if that protein is there, okay? So they looked at the activity of the gene with mRNA, now they wanna look at the protein, and sure enough, look, you can see maybe a little bit of induction of protein for both the silymarin, the S, and the C, the curcumin or turmeric. But clearly where you see the highest level of protein expression of HO1 was with the full formula for Protandim. So does that make sense? So now they've really kind of completed the circle. The gene is on and it's being made into a functional active protein, okay? So if we go down to the next figure, figure 4, this is a pretty cool experiment that they did. So this is that same experiment that they did in figure 1. So using some synthetic DNA and utilizing genetically modified plasmids or vectors. But now what they did was they were utilizing all 5 ingredients, but subtracting 1 out each time, okay? So you'll have the control, and then you have the 5 ingredients minus ashwagandha. The next one, 5 ingredients minus bacopa. The next one, 5 ingredients minus green tea. The next one, 5 ingredients minus silymarin or milk thistle. And the last one, 5 ingredients minus curcumin, and then the full formula there as well. So are 4 enough to activate NRF2? So if you look at panel A, sure enough, we're getting some activation of that light because NRF2 is binding to that promoter and you're getting activity. So 4 ingredients is good, but it's not as good as all 5 together again. So you can see as we're removing 1, you are looking at that activity and you almost work backwards. So for example, at the top, the minus G or minus green tea, right? That's still a pretty potent inducer. And the same for minus ashwagandha or minus bacopa. But where we see the biggest contributions, because it's been taken away, would be the milk thistle or the silymarin or the curcuminoids or turmeric there. But again, it takes all 5 to get the best activity. And they did the same thing that we were just discussing in figure 3 by looking at the protein as well. So activity of gene, and now we're looking at protein levels. So if we go back to figure 4, they're looking, right, to close that loop, and you still see if you lose ashwagandha, lose bacopa, lose green tea, you still get some of the protein being made. But when you remove silymarin or curcumin, that protein isn't as well made. So this protein data really closely mirrors the activity data that they're seeing up above. So again, good proof that it takes all 5 of these ingredients to get that synergistic effect. And when it comes to turning on HO1, we can surmise that it's that milk thistle and curcumin that's maybe having the largest effects in the formula. So if we go down to figure 5, they are looking to see if it is indeed the ARE, that promoter or switch sequence, that is essential to get that activity. So just keep in your mind, you see delta E1, delta E2 in that figure in panel A. So delta means they've removed, and then E1 is a specific region of that switch. E2 would be a different portion of that switch. So Rye, if you can go back to that presentation, I'll try to outline kind of quickly, make it a little more visual for everybody. So if you can go down. Okay, so here we have, you can see down on the far right, we have hemox1, so hemoxygenase 1 or HO1. And then you can see that switch region again. And there are 2 critical components in that switch or promoter region of this gene that they refer to as E1 and E2. And you can see NRF2 is binding to each one of those sequences, but there are also some other proteins that can bind there as well. And this will become important as we start working through some of these other figures. So what they've done is they've deleted, again, using genetic techniques, completely deleted either E1 or E2 or both of them together, okay? So if we go back to that paper, I can walk you through what's happened here. So what they've done in panel A, we'll stay with that figure 5, right? Yeah, perfect. So what they've done, here, drop down one more. So what they've done, you see there you have 15K. So what that means is that's the full promoter or switch sequence, okay? So that's really the control here. Then in that first bar graph, or I should say the second bar graph, they've removed E1. The next one, they removed E2. And then in that last bar graph, they've removed both of those. So you can see individually, there's not a massive effect, because when you remove E1, you still see some pretty good activity. That is a little bit, or it is significantly reduced from the full construct. Whereas if we look at E2, we don't see a significant reduction. So right there, you will say, okay, well, E1 looks to be more important than E2. But what happens when we knock them both out and you see a huge reduction in the activity? So now they know the location in that promoter sequence that's essential for this activity to occur. And that's important because we know that's also where NRF2 is binding. And if we remove one or the other, there's still some compensation that can go on. But when you remove them both, you get a huge hit in that activity. And then just as a kind of a cool control again, so we would just have kind of the salt water for the control in panel B. But then what they wanted to do was look at things that we also know can inhibit the activity, just as some more proof of concept. And so what they've done is just added that synthetic piece of DNA, and they see full activity. Then they've also added KEAP1. And if you remember, KEAP1's job is to bind up NRF2 so that it cannot go into the nucleus. So when NRF2 and KEAP1 are together, they're bound together and we lose a bunch of activity. And then what they did was also inject some synthetic DNA that makes a non-functional NRF2. And again, we lose that activity. So it doesn't matter if NRF2 is knocked out or if it's binding to keep one, we lose that. So this is great evidence, again, that NRF2 and the NRF2 keep one complex is incredibly important for this activity. Then what they did is kind of some cool imaging. These were always some of my favorite experiments to do. You get to play with these fancy microscopes with lasers and everything. But what you can do is, again, grow up your cells in a test tube. And then you can do it live, or you can fix the cell. So usually you'll fix the cell. So you freeze everything in that exact moment you want to freeze everything in. Then we'll utilize those same antibody techniques that have different fluorophores on them. So you can generate these images with all different sorts of colors in them. And what they wanted to do was look to see if NRF2 is indeed going into the nucleus of these cells. I should say the nuclei of these cells. So at the top there, you have control and protandem. And then on the left side, you have what it says Cy3. So that's the actual fluorescent dye that they're using, and it's a red color. So the Cy3 is measuring NRF2. The DAPI, or DAPI, as we call it, actually stains the nucleus or the DNA blue. So now we've got these color differences, red and blue. And what we can do is then overlay the two images to see if the DNA and NRF2 are what they call co-localized together. Right, if you can go to that PowerPoint presentation, I'll maybe clear up what we're looking at here. So if you go up just once, just to orientate everyone, that kind of little bullseye looking thing in the middle of this cell cartoon, that's the nucleus, right? That's where DNA lives without exception. It's bound by membranes, it's a closed off compartment, and it's really selective what it lets in and out of the nucleus. But remember, NRF2 is normally outside of the nucleus in the cytosol or cytoplasm of the cell, and it has to make its way into the nucleus. So by staining both the DNA, we know where the nucleus is in a cell, and also staining NRF2, we should be able to see NRF2 actually entering the nucleus. So Rye, if you can go to that next panel. This was a figure I pulled that is not part of this paper, but I wanted to help you visualize what we're going to look at the figure. So remember, DAPI is the DNA stain. So we can see those are kind of, mostly small, round, blue dots. Then what they've done is use that same red dye to stain something that's in the cytosol or cytoplasm of the cell. But can you see how there's almost a little dark hole in the middle of the red? Mm-hmm. That's where the nucleus would be, okay? So this is clear evidence that whatever protein they're looking at is not making it into the nucleus. And when you merge the two together, that becomes very apparent because the blue and the red are not overlapping. Does that make sense? Mm-hmm. So I did actually copy and paste that figure 6 into this presentation. So this is what they were showing here, okay? So what you can see how at the upper right, protandem, Cy3, you see how NRF2, how the staining has become a lot more punctate. So it's a lot more defined, as if rather than if you oppose that to the figure on the left, where the red is really more of kind of a smear kind of throughout the cell. So it's hard to see where the individual cells are. So right there, there's some evidence that, okay, because all of that red color has gotten really kind of sharp and crisp, it must be concentrated in one region of the cell. Again, they've stained it for the DNA inside of the cell. And when they merge the two together, what do blue and red make when they're together? They make purple. So everywhere we see purple, the colors are basically overlapping. So between the colors matching, the punctate nature of the red coloring, again, pretty strong and visual evidence that NRF2 is definitely being activated and it's definitely going into the nucleus. So compare that bottom right figure to the bottom left figure, and you can see that's much more fuzzy. Things aren't as clear except for the blue staining. whereas the blue and the red very much overlap in that figure at the bottom right. Okay. Does that make sense, Ryan?
Yeah, it makes perfect sense to me. So I'm sure
Okay.
everyone's tracking just fine.
And then we've got 2 more figures to go through. So we've really covered a lot of the background. So we don't have a whole lot to get into for these other figures. But yeah, if you can get me to figure 7. This is a pretty cool experiment that they've done. And this gets to probably speaking to why we see the synergy between the 5 ingredients. So remember, they've only exclusively been looking at NRF2 and its specific binding sequence, that ARE. But they wanted to see if there were potentially any other proteins that are similar to NRF2, if they were also playing a role in activating these NRF2 genes, or specifically HO1. So what they've done is added a whole series of inhibitors. So up in panel A, the control is no inhibitor. So we're going to have just salt water going in there. We're going to try to activate it. And sure enough, we see the HO1 protein being made. Then they started adding different inhibitors for different related proteins to NRF2 to see if they're also playing a role. So they looked at, let's see, 1, 2, 3, 4, 5, 6 different proteins to look at the contribution. And what you can see is that some of these other proteins that are not NRF2 are also playing a role in turning on these genes. And that's important for a couple of reasons. So if you remember, I showed you that E1, E2 figure, and different proteins can actually bind to these different sequences. And so what's happening is some of these other proteins that are not NRF2 are also able to activate the gene. So this is really a good glimpse into the mechanism of how Protandim is working, where this figure is telling me that NRF2 is not the only way that HO1 is being upregulated when treated with Protandim. Okay. This is also probably one of the main reasons why we see that synergistic effect, because these individual ingredients must be activating these different proteins, and then they're all converging into the nucleus to ultimately turn on these NRF2 genes. And then that's just been quantified in more detail there down below in B. You can measure the density of how thick those bands are from panel A, and you can quantify it that way. And basically what they're telling me is that 4 of those different proteins are playing a role in this NRF2 activation. So those 4 significant marks you see there in panel B. Okay, on to the last figure. And this is something I get asked about quite a bit. It'll be figure 8, right? So what about this induction of glutathione? And if you're familiar, glutathione is probably the most important molecule in our body that nobody really knows about. Glutathione itself is a very, very small peptide, so a protein, but rather than being made like a traditional protein or enzyme inside of the cell, glutathione is actually made by 2 enzymes that are regulated by NRF2. And very strongly regulated by NRF2. So for activating NRF2, we should be turning on the genes that are responsible for synthesizing glutathione inside of our bodies. And if that's the case, we should be able to measure an increase in glutathione levels. And so that's exactly what they're showing. And again, looking at a dose response, increasing the amount of those Protandim ingredients that they've added to their cells, we see very much a highly significant increase in the amount of glutathione. And so this is one of those papers where we can talk about Protandim increasing glutathione by about 300%. So
Crazy.
Yeah, and so with that, we just kind of get down into the discussion. I think there's some good points that they put in here. They talk about themselves choosing the right cell culture models to be able to measure the effects on oxidative stress from these two very different cell lines. And again, because they see the same effect in different cells, pretty strong evidence that it is a true biological effect. But there was one line I wanted to call out here specifically, and it states here that, for example, superoxide dismutase and catalase have been shown to be induced. And we talked about that a couple weeks ago. So we know that superoxide and catalase are turned on after we take Protandim. But what's interesting is SOD and catalase do not have that ARE or that NRF2 specific sequence in front of their genes. So the reason that we're probably seeing this upregulation of catalase and SOD is because we know we're also activating these alternative proteins that is what most likely is activating SOD and catalase. And I think that's really kind of the gist of the paper. So really a very mechanistic, a very molecular biology based mechanistic paper that really helps explain how Protandim is working inside of our bodies.
Man, that's crazy stuff. But I don't know about you guys, but that is fascinating to actually look at a peer-reviewed study that way. I mean, oftentimes we read it, but we really have no idea what all of that verbiage is and what, like even down to the chain and the different plugins and how it splits, like the importance of all of that. And, you know, not that we're going to get into that, that much science when we're working with anyone who's interested in obviously consuming the product at any level, consumer or distributor. It's magic, Brian. And, you know, what it does, it also increases our own belief level in what it is that we're doing and why we're doing it. And like Michelle Scaff said, it is powerful. It's powerful for ourselves. It's powerful for the world. And what it does is, for me, understanding, because there's a lot of times I run into consumers that are like, well, I can just go buy this at the grocery store. I can go buy this at the health food store. And you're like, okay, yes, you can. But this, to me, this peer-reviewed study shows that you can't just take certain ingredients and then think that it's the magic be-all for everything. And I mean, we're proving that more and more with Protandim and with our NRF2, that all of these key components and these, I want to make sure I get the verbiage correct on this, but it's the phytochemicals, like how they are all working together and how conveniently that is for anyone who's consuming Protandim for just over $1 a day to have this kind of an impact on our bodies and in our lives is incredible. And so I just can't thank you enough, my friend, on how you just dissect what these peer-reviewed studies are. And now I can go look at that review and really truly understand exactly what it is and why it's doing what it is on the peer-reviewed studies. I know Mary had a quick question. She says, can you share how NAC, which is a precursor, correct, versus Protandim activates glutathione?
Yeah, it's a great question. Oh gosh, if we remember in our NAD discussions and that NAD biosynthetic pathway, we were talking about rate limiting enzymes. And what I mean by that is any biochemical pathway can only work as fast as the slowest enzyme is working. So there's one of these enzymes that's always holding the chemical reaction back. When it comes to glutathione, and there can be rate limiting enzymes and there can be rate limiting substrates. So if something isn't present inside of our cell in high concentrations, you can increase a chemical reaction by just putting more of the building blocks or raw materials in there. So it turns out cysteine is the rate limiting substrate for glutathione synthesis. So it's the amino acid that's the most scarce inside of our bodies. Now, it is easy to get cysteine from the diet, or if maybe you're taking a protein supplement, you should be getting plenty of cysteine from the diet, from your food. But when cysteine becomes limiting, then that also holds back the chemical reaction. So N-acetylcysteine has a lot of great data. So it's just N-acetylcysteine is just a chemically modified version of cysteine that makes it more bioavailable. So if you're limited in cysteine, by taking NAC, you can potentially increase your glutathione levels. But what is the most limiting is actually the rate limiting enzyme. And it turns out it's the first step in putting the first two amino acids together in glutathione that limits the reaction. So the best way to increase your glutathione levels would be, of course, to make sure you're getting enough protein in your diet, and especially cysteine containing protein, but then also utilizing these nutrigenomic techniques to increase the enzymatic machinery so that we can actually make more glutathione. So we can see in this study, they didn't add cysteine to the study or N-acetylcysteine to the study. All they were utilizing was protandem, looking at the nutrigenomic effects, and that was enough to get the induction of glutathione.
Wow, that's incredible. That is super cool. Let's see what Stephen has. Says, I'm still intrigued by triggering my own luciferase activity to be able to luminesce. Could this be a new product that we could develop? Let your light shine. I love that, Stephen. We should come up with that, even on a shirt, like let your light shine, and just boom, the luminescence.
Yeah, that's really funny, actually, Stephen. But yeah, there have been studies where you can put these different fluorescent or light emitting enzymes into organisms and they will actually glow in the dark.
Wow.
So careful what you wish for, Stephen. That stuff's coming.
Well, guys, you've been fantastic. Thanks for working with us. I know we're an hour and 20 minutes. That's a longer version than normal, just with some technical difficulties. Thank you for staying with us. We will get this recorded. We have it recorded, but we will get this up in the Vision Summit YouTube channel. Refer to it, use it, listen to it over and over again. Just increase your belief, guys. This is what this call is all about. Not only knowledge as to why and what and how we're taking it, but just to increase your belief levels on exactly why this company is so important for the world and where we're taking it and where we're headed. So I love you guys. I respect you. I appreciate you. I praise you. Brian, thank you for your knowledge, man. Thank you for being so willing to share. You know, you've got a huge amount of work that you've put in from study time, you know, take 20 years ago and just compound all of that study, that knowledge, that sacrifice of just, you know, looking through text and understanding what this whole world is with regards to the scientific level and sharing it in such a layman's way for us to understand. So we look forward to it, guys. I'm going to sit down with Brian this week. We're going to actually map out probably about 4 to 6 weeks out so we can promote better. You guys will know what the topic is from week to week. So you're not just showing up hoping that it's a benefit. It'll always be a benefit, but you know what I mean? Just to kind of, for you guys to do a deep dive on the week subject before. So we'll do a better job of getting what those topics are out probably 4 to 6 weeks out. So Brian, thank you so much, buddy. I appreciate you. And thanks everyone. Go have a great, amazing day. Take care.
Thanks, everybody.